thank you for joining us today. We're here to hear about a very interesting company called Biointelli. And we have the pleasure of speaking with David Hines. David, you want to tell us a little bit about Biointelli?
Well, thank you, Dale, for this opportunity and this recording. Biointelli, actually, its origins started when I had left the lab some 25 years ago and wrote a book after several years in marketing called Focus Marketing, Focus Selling Using Government Databases back in
Hmm.
the early 90s. And I had brought the technique and strategy of using publications for direct marketing and sales using
Mm-hmm.
publication conferences and funding.
Mm-hmm. I see. And I see here, right, founded in 2008, and you were very much at the forefront of bioscience marketing. What can you tell me about that?
Well, it evolved from the book of using government databases to sell life science and biomedical science into an evolution of when the government databases came online. The CRISP, the computer for retrieval for information on scientific projects, came online. We made these simple scrapers and curated the data. And that's how it started. Companies immediately saw the value of using this new digital data to do word searching and direct marketing
So there
for the.
wasn't any kind of API or ability to get the data directly. You had to actually pull it off of the screen. Is that what you mean by screen scrapers?
That's correct. Screen scrapers, they were just screen scraping, and there was no way the government didn't offer any export or any APIs as they do now. It was strictly screen scraping, and there was no information. First name, last name, institution, state, and country. That was it. We had
And
to
so
go
these
figure.
were these were like RO1 and different types of NIH grants. Is that correct?
Correct. And we wrote supplemental material to explain to everybody what's an RO1, what's an S10 grant, what's a clinical grant. They all signify different things for sales and marketing.
I see. And then how would the sales and marketing professionals use this information?
Just as we started preaching, basically bringing this new funding, and that's what we became quite famous for, was the newness of the data. So we would track new funding as it happened, and then it started evolving beyond government databases into venture capital, foundations, different funding agencies, then international funding databases, the DFG in Germany, the Kaken in Japan. And it kept just snowballing and evolving into more and more data, digital data.
So now you've got a new version. Do I understand that correctly? About what is it? You call it Biointelli or excuse me, Biointelli 2.0?
That's correct. For the last 16 years, we had siloed databases, databases inside of databases, in which we had funding databases where scientists in that bucket would be listed there. And then we had abstracts and publication databases for that, PubMed. And then we had conferences databases for non embargoed or open access information that we would curate, pull information from, and create another database. 2.0 was the effort to unify or bring together all of the segmented databases that have been that way for a long time and add new features to it. And that's what we did in 2.0.
So before, I understand that the databases, what you just said, were individual like silos. Is that correct?
They were siloed. The
Yeah,
information did not talk to each other.
I see.
Grants, publications. So you could have somebody who published a paper this week or this month, and he might also have a grant. But and then the issue was they were moving, constant moving target of scientists jockeying around from postdoc positions to tenured PIs to assistant professor to full professor. So it's literally like shooting a moving target. So on the unification, it solved several problems of basically keeping track of the scientists. Then we decided, after looking at the market, to say, why don't we create the arc of a scientist's history from their graduate level all the way to their recent position in industry, if they went that way, or academia? And that's what we've now created in the
So you
2.0.
can follow an individual from their graduate school papers through when, say, they received an assistant professorship somewhere, different location, received a couple of grants, and then say moved into industry and you still follow them. Is that correct?
Correct. Yeah, and we update their contact profile as they arc through time.
I see. So, as
Okay.
far as Biointelli 2.0, who is the intended customer for this kind of service?
In the beginning, it always was life science industry. The life science companies, Kyogen type companies, Biorad, Thermo Fisher, all of the companies selling products into the basic research market. And then it had started to expand as well into diagnostics. Now with the biopharma database, diagnostics, CROs, CMOs, contract research organizations, contract manufacturing organizations, and again, more of an evolution.
Hmm.
A lot of the public databases include just the publicly known funded project with no information on methods and materials. And that's really what a lot of these life science companies want to know. What
Right.
products use in that grant? We've appended that now in millions of grants, all of the methods and materials sections to that.
So, say if I'm an HPLC column provider and a very specific kind of purification preparation in front of mass spec, then you can go ahead and actually find out which investigators have published using that particular, say, trademark brand from the materials and methods.
That's correct. So if you're interested in a particular anion exchange column, con A column, whatever it might be, or a particular piece of mass spec equipment, you can now reference that keyword search into the grant and find out instead of looking at the grant and then going to a link that says here's the papers that were attributed to this funded project, and then seeing through the methods and materials section what exactly they used, you can now circumvent those steps. We've basically consolidated steps to save time and money and consolidated over 2,000 different funding organizations, millions of publications, and conferences that have been appended, and we've curated and researched the data. And that's another evolution that started to basically study the conferences and basically if somebody was attending, we go and research um, products that were mentioned and then turn over to the company that information. And that's how the conferences started.
So then, for your audience, by this I mean the users of BioIntelli, is going to be people that are interested in digital marketing, basically looking for leads. They're looking for suspects in order for them to hit them, message to them, right? Some particular offer about something that's of interest to them.
Correct. Some of the biggest uses are, let's say, tracking older models of equipment. So you can actually drill down into model numbers and particular from capital equipment that on old mass spec equipment to old HPLC waters equipment or an ABI 7500 old workhorse PCR equipment that's
Mm-hmm.
still being used. Product conversion and even looking at contemporary kits. It's a very common tactic. People want the box model, the box dream, where the box costs $35 to make and you can sell it for $300, the Kyogen model type situation, where they develop a better box and then want to try and convert. But what they need are large lists of people who are using that box. And to find that, you have to really go through the methods and materials section
Yep.
and consolidate all of that data, their emails, their phone numbers, and then put that into inside sales or digital marketing department of
Mm.
marketing.
So, would you like to go ahead and start giving a little bit of a demonstration of the
Yeah,
platform?
absolutely.
Okay, let's go ahead and take a look.
So the main login, we have the main database where we actually have two programs now. One is a standalone to assist in sales, and then this is Biointelli 2.0. With 2.0, it is now a Boolean logic setup where you can add complex search queries and or not, and further refine your search. So if you're looking for CRISPR and mouse or a particular cell line, human, or CRISPR and any other scientific methodology to refine your target group, Some of the downsides with current marketing methods is it's not specific enough to do highly technical personalized outreach, and that's what you really have to do. But let me give you a quick version of 2.0. So this is the home screen. You basically use a keyword. What's changed now is we've consolidated using Elasticsearch, including the US government's clinical trial database, for grants. So here we have 104,000 CRISPR grants, the new gene editing technology. We linked to 21,000 conferences and 272,000 publications. So welcome. This is, thank you, Dale. This is Biointelli 2.0. So here we did a search for CRISPR, the new gene editing technology. And we've added many new features to the 2.0, specifically a Boolean complex logic to refine your search. So let's say you were looking for CRISPR in mouse, CRISPR in rat, CRISPR in a particular cell line, CRISPR in another scientific modality to further refine your group. Because really, what you want to do in life science marketing, you have to speak directly to the scientist for direct scientific marketing. You have to be refined as possible. And that's why programs like Zoom don't work. There is not enough granularity in their data to do, let's just say, palpable outreach to scientists who know that somebody just wrote immunology
So
or
what you're
molecular.
referring to is Zoom info, to be
Correct.
clear. You said
That's correct.
Zoom, and I
Oh,
got
I thought
confused.
everyone had Zoom info. Zoom
Yep.
info, a formidable database, but not enough granularity in the database to be formidable tool, and not enough information cross-referencing all of the other information that's cross-referenced in our new database 2.0. So with this, you can do very complex queries to refine your search, and you can also go into organization name. In the old database, Biointelli, you used to have to use the series of wildcard functions. People probably remember this. There's people that have been using Biointelli for well over almost 15 years, 16 years since we digitized it and formed the company in Massachusetts. And basically, it was a complex game to, let's say, get everybody at the University of California at Davis. You used to
Mm-hmm.
have a series of complex query structure. That's been alleviated. We also had an issue with naming countries and states and organization. Now we can zero in on it by doing things like saying South Korea, South Africa. They're all pre-programmed, and you can even go into states. So if you wanted to put in United States, you could just pick the United States. And then once there, you can get further refinement by
Hmm.
going into country. And it's all pre programmed. It
If you
wasn't like
wouldn't,
that in the old database.
if you
It
wouldn't
was,
mind, then the terms that you had in the upper left hand corner, I can't read. What you, if you can go ahead and include the terms, and then what do some of these lines mean?
oh, sure. So here we did CRISPR, and you can select your functionality, or you can deal with it directly on the little pop-up screens. You can say include all companies that mention CRISPR in their tech stack. You can say I want to see most recent publications, conferences, grants, clinical trials. And again, many of the conferences come out of embargo after 6 months, and it's open access information and also licensing, which we're very big on. We've always been very strict on ethics, licensing, and permissions. And you can further either drop and say, I'm not interested in companies, or I'm not interested in publications, or just grants. So here we have everything selected, and you can then pivot through the data and see every individual scientist. So if we picked on this Elizabeth Simpson at the University of British Columbia Medical Genetics, I'm going to share her tab now. Basically, the front horizontal bars showed basically the same information, where we list and link to her latest conferences, grants, and publications. So we can see October of 2023 she has a recent conference that we can click on and go to them directly to their website. So we're linking the data to the conference,
So
and you can see it from her presentation at the poster.
so this was an investigator in CRISPR that you drilled down into, and you found her particular presence at a scientific conference. And here you're pulling up the actual abstract.
Correct. We have sophisticated crawlers that are linking all of this data. Now, what we've done is it talks back to the database to
Mm-hmm.
basically curated all the information that is not in the public domain. Her phone number, her email address, and her entire, going back to what I was saying before, even further back, going from her graduate school all the way to
Mm-hmm.
her recent positions, where she's held positions, her activity, basically the arc of her life, of her contributions. So we can see now it's all consolidated. Her grants, her publications, her recent conferences are now all tracked, including clinical trials, which she doesn't have any here. And we can see everything from 2023, her most recent grants. She has three current grants. And we can just click on it and see her latest funding using CRISPR technology. And now we
That's
know
great.
that she's involved with gene therapy to prevent probably macular degeneration, and she's using CRISPR technology to further that study.
So
So
if
this is the
mhm.
new format that's been done.
So if I have a person, then I wanted to find out more about, basically, I know you searched under CRISPR, you found a particular individual, you found the conferences they were at, their funding, their publications. Then, as a sales rep or as a marketing director, you can use that information for tailored outreach. Is that correct?
That's correct. That is correct. Now, that will lead into our second application, our new product that we've developed. We've done a deal with OpenAI to basically marry OpenAI's technology with this data. We call it BioConnect Pro, and we'll touch on that at the end of this, interview here. But what I'd like to do is now some people say, well, that's great. We have their grant. You can see my screen, I assume. Yeah, we're still
Mm-hmm.
showing. We can see, yeah, we have grants, we have this. Now, what about companies? We're interested in academia. We're interested in industry. We want to see, what companies are do you have that are doing CRISPR technology, and what
So this
about the
it might be say a CRISPR enzyme supplier or a guide RNA supplier, and they're looking for companies that are sort of doing CRISPR therapeutics so they can perhaps sell their OEM services, something like that.
Well, you could do quite a bit here. You could, so right now I'm on the industry tab. I opened up CRISPR Therapeutics. You can see that, correct?
Mm-hmm.
You could, okay, so you can see this. Now, exactly. Let's say before, instead of using LinkedIn Navigator or cruising through LinkedIn and saying, because LinkedIn Navigator is very limited for contact details. Sure, you can buy programs like Seamless AI, and you can, or Apollo, or even Cognizant, and you can say, I want to latch on, and I want to view, uh, their LinkedIn names, but then you have to go and find their phone number and their contact details. Here we've consolidated, again, this is the Biointelli methodology, consolidating the information. And we categorize it for you. Up here, we have they're classified technically as biopharma, biotechnology, and genetics company.
Mm.
And we can go through basically an overview of the company's tech stack. We also include the general email, a link to their LinkedIn profile, the main
Mm-hmm.
company member, their Crunchbase link, and their recent funding. What are they up to? 127 recent projects. But we also link to their most recent conferences. So
No.
you can actually reach
Mm-hmm.
out using our information to get to their most recent presentations
Mm-hmm.
um, and the keywords that are also being mentioned in those conferences.
Okay.
So here we can see we could do this for Merck, we could do this for Genentech, we could go to everyone, or we could go back and say, I'm interested in talking to their director of clinical affairs or their
Mm.
senior director of genomics and computational biology. You can separate them here, or scientist one, or scientist two, or head of technical
I
operation.
see. Like an
All of
account based marketing sort of campaign where you're targeting different people at different levels of the organization. That's
correct.
super. Well, it's obvious that there is a lot of functionality you've built into this. A question that I have for you with Biointelli, as far as being able then to export this data, I assume then it has different connectivity in terms of exporting the data into different flexible formats.
No, absolutely. We're in the process of finalizing our Salesforce App Exchange app that will interface with Salesforce. We're just
Great.
waiting for the approval process from Salesforce and their security vetting of our Salesforce app. It's being done as we speak. We also have developed over the last decade, which has basically been the backup for the Salesforce is APIs custom made for each of our clients, and
Mm-hmm.
also FTP CSV concatenated files that
Mm.
they've custom tailored to us to say, instead of getting the information in this format over here, you can export all of this data and all of the contacts with
Mm.
their emails and phone numbers directly here.
Thank you, David, really, for your time here. It's been super interesting, right, to see the power of what BioIntelli Pro can do. How do they
This
get a
is
hold of you?
2.0. This is the 2.0. We
The
haven't even gone.
2.0?
Yeah, we haven't gone to the Biointelli Pro yet.
Oh, okay. So the BioIntelli 2.0. I guess for getting in touch with you, the best way would be probably by email. Is that correct?
Sales at biointelli.net or the office in Cambridge, 617-444-8420 is our direct line.
Very good. Well, thank you very much for joining us today. This is really interesting.
Thank you, Dale.